This tutorial will focus on meiosis, and perhaps rather than writing out the process in full, we'll just have a look at this diagram here, which I think sums it up very nicely. And there are lots and lots and lots of diagrams you can find on the internet, in books. There are multiple animations you could have a look at on YouTube to get your head around the process of meiosis. But this one will at least help outline the steps fairly clearly and show you what is going on. So remember that all of this happens in the context of the cell cycle, and that we're talking about cells that are found in the reproductive organs of organisms. In humans, that would be the ovaries and the testes, and the germline cells, the spermatocyte and the oocyte, will go through the process of meiosis to form 4 new sex cells. And the thing about the sex cells is that they're haploid. They contain only half the number of chromosomes compared to the original parent cell. So it's during interphase, and remember interphase is the entire part of the cell cycle other than the meiotic division. So during interphase, and in fact the S phase of interphase, the chromosomes will replicate and make exact copies of themselves. So by the time we see the chromosomes as visible at the beginning of prophase 1, the chromosomes have already replicated and will appear as a pair of sister chromatids, noticed here, joined by a centromere. They will come to lie next to each other physically as a homologous pair of chromosomes. So there'll be one that has originally come from the maternal side and the other one from the paternal side. So a pair of chromosomes, so a homologous pair, once each is replicated into sister chromatids, is described as a tetrad. At the same time, early in prophase, the spindle will form. Spindle fibres will form between the centrioles, and some crossing over and recombination may also occur randomly at different parts of homologous pairs. It could happen multiple times down the length of the chromatids, or it might only happen once, or it might not happen at all. The actual crossing over point is described as the chiasma, and if there are multiple points, it would be described as chiasmata. So this is crossing over and recombination. Late in prophase, the nuclear membrane will break down and disappear, and the chromosomes will attach to a spindle fibre via the centromere. In metaphase 1, each pair of chromosomes, each homologous pair, will line up down the equator, or the centre of the spindle, shown here, the metaphase plate. You can see that in meiosis that a homologous pair of chromosomes is attached to the same spindle fibre. And this is different to mitosis. In mitosis, each chromosome or pair of chromatids would attach to a separate spindle fibre. But in meiosis, a homologous pair joins to the same spindle fibre. In anaphase 1, each member of the homologous pair will be pulled back by its spindle fibre to the opposite pole. So this is one member of the homologous pair, still existing as a pair of chromatids, and this is the other member of the homologous pair, still existing as a pair of chromatids, and each is pulled back to opposite ends of the cell, and likewise with the other homologous pairs. Then telophase 1 will see the formation of 2 new cells, the reformation of the nuclear membrane, and at this point each cell now only has half the number of chromosomes compared to the original parent cell. So in this cell that's shown in this diagram, the total number of chromosomes is 4. By the time you get to the end of telophase 1, we're down to each cell having just 2 chromosomes. So this is one chromosome, and this is the other chromosome. Each chromosome is still made up of a pair of sister chromatids, but whereas there are 4 chromosomes here, there are now only 2 in each cell here. So that halving to the haploid number has happened by the end of the first division. Then each of these cells, so this cell here and this cell here, will now proceed through a second division of meiosis. It's important to realise that this second division is identical to what happens in mitosis, but it's very important to realise that you don't call it mitosis. It's not mitosis, it's not a mitotic division, it is in fact the second division of meiosis. But in terms of learning what actually happens to the chromosomes, it's identical to what happens in mitosis. So the cell here will go through prophase 2, where the nuclear membrane will break down and the spindle fibre will form. Each pair of chromatids, that is each chromosome, now attaches to a different spindle fibre, so one here and one here. Metaphase will see them line up down the metaphase plate or the equator. At anaphase 2, the chromatids pull apart, so the centromere divides, the chromatids pull apart and move to opposite ends of the cell. And then in telophase 2, 2 cells form from each cell. The nuclear membrane reforms, and each cell, which of course is a gamete or sex cell, now has 2 single chromosomes present. This of course happens to the other cell that's formed during the first division of meiosis, and exactly the same process takes place to end up with the other 2 cells. So altogether we end up with 4 cells, each of them haploid, so each of them with 2 chromosomes in this case. So let's have a look now at how this relates to the formation of sperm and egg. So this diagram's not a bad representation of this, and again it just tracks you through the process we've just been through. So your germline cell, your starting cell in males, is a spermatocyte, which is diploid, and then goes through a meiotic division. So there's the first division of meiosis to form 2 cells, each with half the number of chromosomes compared to the original spermatocyte. Here's the second division of meiosis. It sees the formation of 4 cells, spermatids, which then get modified into sperm cells, which are the gametes. These, of course, are haploid. In the female, you start with the oocyte. Again, first division of meiosis. Now, what's interesting here is that by the end of the first division, the cells that are formed, one of them is fully formed, the other is only partly formed. It's a kind of a reduced size cell. Each of these cells then goes through the second division of meiosis, but this cell here, which isn't fully formed, actually produces 3 inactivated cells, sex cells, called polar bodies, and then you have 1 active egg cell or ovum. And this is the 1 cell that remains in the ovaries, which will then be available for possible fertilisation. So of the 4 cells that are produced, only 1 is active and 3 become polar bodies, which are inactive. Here's a lovely photograph taken under an electron microscope, by the looks of it, of anaphase. Let's have a quick look at some of the key differences between mitosis and meiosis. So, in mitosis, 2 daughter cells produced, each of them diploid, same as the parent cell. In meiosis, 4 daughter cells produced, each of them haploid, that is N, and that is half the number of chromosomes compared with parent cell, which is diploid. In mitosis, during prophase, the homologous chromosomes do not line up with each other, do not lie next to each other. In meiosis, the homologous pairs, or the homologous chromosomes physically lie next to each other. This often leads to crossing over and recombination. Between sister chromatids. In mitosis, a pair of homologous chromosomes attached to the same spindle fibre. Whoops, my mistake there. That is meiosis. Sorry, so pair of homologous chromosomes attached to separate spindle fibres. My mistake. So each member of the homologous pair attaches to a different spindle fibre. In meiosis, homologous pairs attach to the same spindle fibre. Alright, so we'll see something like this over in mitosis. We'll see this. In mitosis, the chromatids pull apart during anaphase, but in meiosis, the chromatids only pull apart in anaphase 2. So in anaphase 1 they remain connected. So these would be the key differences between mitosis and meiosis. And of course we might say that the final purpose of each, so the purpose of mitosis is for growth, the growth of an organism, that is, so where new cells are needed, cell division needs to take place, and so growth occurs, and also the repair of damaged tissue or tissue that needs replacing, like blood. So mitosis will replace damaged tissue. In meiosis, the purpose is to produce sex cells or gametes, which is their proper name. So the purpose is to produce sex cells which are haploid, so that during fertilization two haploid gametes will combine to produce a zygote, which again is diploid. In mitosis, of course, you have a parent cell which divides into two daughter cells which are both diploid as well. And then these cells will divide and produce daughter cells that are diploid, and so on.